Okay, this chapter is a male reproductive system. We're going to look at the sexual reproduction and development. It says in there we have two sexes. Obviously, in the case of humans, there's males and females. And next topic is overview of the reproductive system. As you can see here, we have primary sex organs, right, and secondary sex organs right here. Secondary sex organs. Primary sex organs are the ones that we are born with, such as, for example, in the case of females, the uterus. In the case of boys, the penis. And secondary sex organs are the ones that we develop when we reach puberty, such as, for example, breast enlargement in the case of females, and what we call the Adam's apple gets enlarged in the case of males. Also, we have obviously external and internal genitalia, right? In the case of, for example, females, internal would be the vagina, for example, and external would be the breast. So that's fairly easy to understand. We keep scrolling down. You can see in there androgen insensitivity syndrome. That's a disease. We are not going to cover that at this time. So chromosomal sex determination, obviously, that is determined. The gender is determined by your when the spermatozoid and the ovule get in contact and fecundation happens. The ovules can be XX, or the spermatozoids can be XY. That depends on the chromosomes. We have 22 set of pairs which are called autosomal chromosomes, and we have one pair that is going to be sex chromosomes. So if the combination of the spermatozoid and the ovule is X and X, as you can see right here in the female, I mean, you're going to get a female, as you can see right here in this figure. And if it's X and Y, then it's going to be a male. Okay, so we keep scrolling down. Hormones and sexual differentiation. One thing that you need to know, or you need to understand, is obviously the gender determination happens immediately, right, when the spermatozoid and the ovule get together and fecundation happens. But the cells start dividing and a new human starts growing inside the mom's uterus. But up to 5 to 6 weeks, as you can see here, the embryo is not clear whether it is female or a male. However, as weeks go by and they start passing, you're going to see here that this is now a little bit more clear. It's 7, 8 weeks right here, 8 weeks around here. One thing that you need to pay attention to is that these cells that you have right here at the level of the abdomen, you see even the colors, blue and orange, they are going to be the gonads. So in this case, if this is a male, this is going to be the testicle, and if this is a female, these are going to be the ovaries. So if we scroll down, we can see right here that the ovaries, move from the abdomen to the pelvic area, not much to travel. But in the case of the testicles, they travel from the abdomen all the way down to where the scrotum is going to be. So there's a lot of areas that the testicle will have to go through in order to descend to the scrotum. And this happens obviously from 7 to 8 weeks until the baby is born. Okay, so you can see that in here the structures are already very much in place when the baby is born. So if we scroll down, external genitalia, same thing. 5, 6 weeks is not clear what it is, but around 10 weeks, you can say 8, 9 weeks, you can see right here that in the case of females, it's going to start, this is going to remain opening, right, which is going to turn eventually into what is going to become the vagina, labia majora, labia minora. But in this case of the male, this area is going to close. This is going to elongate in order to form the eventually what the penis is going to be. Okay, so we keep scrolling down. Descent of the gonads, again, is very important because, as I was telling you, the cells that are going to turn into gonads, testicles and ovaries, they need to descend, in particular the testicles. And as you can see right here, testicles are going to descend basically from the abdomen down to where the scrotum is going to be, and that is promoted by the presence of testosterone and some anatomical structures that are going to be open so the testicles can actually descend. We keep scrolling down. There's nothing else after that. One last word. Cryptorchidism is the term that is used when the testicles get stuck in the middle of the pathway between the abdomen and the testicles. In these kids, the scrotum is not going to show a testicle. Okay, the next topic is male reproductive anatomy. We see in there the first part, which is going to be the scrotum. You can see here the scrotum right here. This is like a bag, like a pouch, where the testicles are going to be located. And we have actually two, because we have two testicles, and there is a separation. There is a wall that is going to separate both testicles. And you can see right here, this is the perineal area. Okay, that's why this is called the perineal raphe right here. Okay, so that's where we have the scrotum, and inside the scrotum is where the testicles are going to be located. So I was telling you, there's a wall in the middle of the testicles, which is called the median septum, and the perineal raphe is the way you see it, is the outside view of the median septum. The left testicles are, one of them is a little bit lower than the other testicle due to the fact basically to stop or to avoid both testicles to be hitting each other. So if we scroll down, we have in here the male reproductive system, and the function of the male reproductive system is basically to provide the spermatozoids in the semen in order to have a baby. Therefore, the main idea is to have the spermatozoids coming out of the penis. So where the spermatozoids are going to start? The spermatozoids are going to be created right here in the testicles. The testicles is just a set of tubes that is coiled, forming this mass, which is going to be the testicles. So it's a set of tubes, and the first part of that tube is going to be the seminiferous tubules. The spermatozoids are created in the seminiferous tubules. So a set of tubes that are going to come out of here, the tube is going to continue up here through the ductus deferens. You can see that right here. This is the ductus deferens. The ductus deferens keeps going up into the body, goes around the urinary bladder, goes down into the penis, and then it comes out through the glands. So the spermatozoid will come out. Okay? So one thing to look at is that this is the testicle, and it's basically far away from the body. This is the body, right? And the testicles are hanging in there. So it's separated from the body. But the testicles are a group of cells like any other part of the body. Therefore, it needs to have nutrients. And that's why we have right here blood vessels. Also has a lot of sensitivity. Therefore, it has to have in here a lot of nerves. Not only that, the testicles move up and down because of the presence of muscles, such as the cremasteric muscle right here and the dartos muscle, as you can see here, dartos muscle. Okay, these muscles are going to help maintain the temperature in the testicles, because one very important characteristic is that the spermatozoids are not going to be viable. They're not going to survive if there are changes in the temperature. The temperature must be always the same in the testicle. It cannot increase a lot or decrease a lot, because the spermatozoids will not work. So for that reason, when the temperature is too cold outside, that muscles are going to constrict and the testicles will move closer to the body to increase the temperature. If the temperature is too hot in the body, like for example when somebody is performing physical activity, then the testicles move far away from the body in order to remove the temperature from the testicles. The blood, the ones that go through the arteries, is hot because it comes from the body. And that would be too hot for the cells in the testicles. So therefore, the veins, as you can see here, these veins right here, that are going to form the pampiniform plexus, they're going to remove this heat and they're going to bring it back into the body. So since you remove the heat from the blood that you have in the arteries, then the temperature in the testicles will not increase that much, therefore allowing the spermatozoids to be viable. So let's keep scrolling down. Right here we said the cremasteric muscle and dartos muscles. We already talked about that. Pampiniform plexus, we said is to remove the temperature. And here you can see, red warm blood, cool blood. And you see here this blood is going to the testicles, but the temperature in this blood gets removed by the pampiniform plexus. That's why in here is becoming colder. And this pampiniform plexus goes back to the body. So let's keep scrolling down. The testis, we already mentioned that is inside the scrotum, but if we cut the testicles, we can see here the set of tubes that I was telling you is just a cold set of tubes. I told you that everything starts with the seminiferous tubules that you can see right here. That's where spermatozoids are going to be created. Spermatozoids are created in the seminiferous tubules. The seminiferous tubules continue into the retitestis, which is this area. The retitestis continues into the epididymis. This is the head of the epididymis. Goes down into the tail of the epididymis, and from there it goes up into the ductus deferens, as you can see right here. This part right here is called the spermatic cord. That is kind of like the bridge that communicates the testicles with the body, which is the same area that I showed you before in the previous figure. So this area right here is gonna be the spermatic cord. So what do you find in the spermatic cord? Blood vessels, nerves, ductus efferens, muscles. So a very important structure because it allows the spermatozoids to reach the body as well as the blood. So you can see right here all the tubes and everything. The part of the tube that is going to take the spermatozoids out into the penis is going to be this tube, which is going to be at this level, the ductus deferens. So where the spermatozoids are created in the seminiferous tubules, and it continues all this set of tubes and comes out through the spermatic cord into the body using the ductus deferens. What are the structures you find in the spermatic cord? We said, besides the ductus deferens, we said blood vessels, arteries and veins, as well as nerves. Okay. So if we scroll down and we cut one of these seminiferous tubules, this is the tubule, and you can see here a whole bunch of balloons in there. These balloons are the germinal cells or germ cells. These cells are the ones that are going to produce the spermatozoids through a process of meiosis. So as you can see here, these cells are going to start going through a process of meiosis. In meiosis, you have two cell divisions. Therefore, you're going to have first cell division, second cell division, and then in the middle, they're going to basically the spermatozoids are going to fall in the space or in the lumen that the tubes have. And you can see here they're showing these are the tails of the spermatozoids. You have this similar figure in here where you have the tube. These cells in purple are going to be the germ cells or germinal cells. They go through meiosis in order to form the spermatozoids, and the spermatozoids are going to fall in the lumen in the space of the tube, ready to come out into the penis. So if we keep scrolling down, we can see here the spermatic core, the spermatic ducts. We already talked about that, but we can see the same figure in here again. So we mentioned that right here in the testicles, in the seminiferous tubules, that's where the spermatozoids are going to be created. They go through the retitestis, epididymis, head, epididymis tail. Ductus deferens goes up, as I was telling you. It goes up, goes around the urinary bladder, and it joins the seminal vesicle, forming the ejaculatory duct, and the ejaculatory duct goes through the penis, as you can see here, and the spermatozoids are able to come out. So if we keep scrolling down, you can see the same figure in here. Again, we have right here the testicles, the seminiferous tubules, that's where the spermatozoids are created, epididymis, retitestis, head of the epididymis, tail of the epididymis. We have the ductus deferens going up, goes around the urinary bladder. Okay, then the tube, the ductus deferens, and the tube from the seminiferous tubule merges in order to form the ejaculatory duct. It goes inside the prostate. It continues through the penis in order to come out right here. Okay, in here you can see the glands that are part of the male reproductive system. We have seminal vesicles, prostate right here, and bulbourethral gland. The spermatozoids, together with the fluids from all these three glands, are going to form the semen. The semen contains 60% of the fluid from the seminal vesicle, and 30% of the prostate, and just a little bit of the bulbourethral gland, as you can see right here. As spermatozoids obviously are going to be basically 5 to 10%. So we keep scrolling down. We have the penis. The penis is just the structure that is going to allow to the spermatozoids to be placed, or semen I should say, to be placed in the vagina. You can see here it has a shaft and a base, and it has these structures right here that are going to be like sponges. These sponges in here, they are called corpus spongiosum right here, corpus cavernosum right here. They fill with blood, and that is going to make the penis to become very hard. Blood contains water. Water gives you pressure. So the more blood you put in here, more pressure you're going to have, and that penis is going to become very hard. So right here in the corpus spongiosum, you see the urethra. That's the tube that is going to be used in order to allow the spermatozoids to come out. The urethra is also part of the urinary system, and obviously is used for urination. And that's about it.